Hello everyone. Today we are going to discuss the affinities of Balanoglossus to Echinodermata or phylum Echinodermata. In affinities we discuss the similarities and the dissimilarities of these Balanoglossus with the phylum Echinoderms. What are the adult resemblance? Adult Balanoglossus and Echinoderms are structurally quite different. Why? This Balanoglossus showing the what? The biradial symmetry, and Echinoderm shows the radial symmetry or pentaradial symmetry. So it is difficult to draw their phylogenetic or evolutionary relationship between themselves. However, some resemblance between these Balanoglossus and Echinodermata are as follows. First is the what? The origin of coelom. In both, that is Echinoderms and Balanoglossus, the coelom is the enterocoelic origin of the coelom. What do you mean by this enterocoelic? When there is a cleavage and the blastulation occurs, after blastulation, the cavity appears. That is the what? This blastocele, and after that the gastrula forms, and this blastocele reduces and new cavity appears, that is the archenteron. And after that archenteron, the three germinal layers are formed. And during that, there is a formation of the coelom. If this coelom, this is the what the coelom, there is a early mesodermal pouch, and these pouches are going to form. And this is the what? The embryonic gut. And from this embryonic gut, if this coelom forms, then we call it as an enterocoelous type of the coelom. So, in both echinoderms as well as the Balanoglossus, the origin of the coelom is the what? Enterocoelic type of the coelom. The gastrula elongates along the what? Anterior posterior. This is the anterior and the posterior axis, and anterior tip of archenteron is differentiated into the coelomic vesicle, and that is called as a what? This protocoel. And thus origin of the what? This coelom is a what? The enterocoelic. This is the formation or the origin of the coelom in the what? Balanoglossus. This is the enterocoelic type of the what? That is the coelom that is found in both the organisms, that is the echinoderms as well as the Balanoglossus. The remaining posterior part of the archenteron, it marks the future gut or the alimentary canal. So if it develops from the what? These the gut part or the what? That is the embryonic gut, then the coelom is the what? Enterocoelic. And enterocoelic type of the coelom is found in Balanoglossus as well as the what? Phylum Echinodermata. Then the second affinity or the second similarity in the adult of echinoderms and Balanoglossus, that is heart vesicles and glomerulus of the Balanoglossus. That heart vesicle and the glomerulus, where it is present in the circulatory system, and this is the heart vesicle, this is the heart vesicle, and the what? The glomerulus of this, of these Balanoglossus, that are considered to the homologous to the dorsal sac and axial gland of the what? Echinoderm. So in echinoderms, the what? This heart vesicle as well as the what? The glomerulus, which is, it is considered to be the what? Homologous to the what? The dorsal sac. This is the dorsal sac, and this is the what? The axial gland. So this is the homologous to the dorsal sac and the what? This uh, dorsal sac and the what? The axial gland of the what? That is the these echinoderms. And this heart vesicle and glomerulus of these Balanoglossus is comparable to the what? This dorsal sac and the what? This is the axial gland. If you observe here, this is the heart vesicle, and this what is the function of this dorsal sac? It acts as a what? That is a pumping organ or the like the what? That is a heart, and this is the what? The axial gland. Here the what? That is a heart vesicle. It acts as a what? Pumping organ, or it acts like a heart, and the what? That is a glomerulus. This glomerulus, it is where it is present toward. This is the what the glomerulus you can observe here, and this glomerulus. So these are the what the comparable structure or the homologous structures which are found in both the groups. Then the third affinity, that is the nervous system of Balanoglossus and Echinodermata. This nervous system, this is the Balanoglossus nervous system, and this is the what? The echinoderm, or you can say that is the what? The nervous system of the sea star or Asterias. And if you observe that there is a no brain as such, these here you can observe the what that is a these cords or the nerve cords and fan-like nervous expansion. Then this is the collar cord. Then the circumenteric nerve ring, ventral nerve cord, dorsal nerve cord, like that the what the nervous system in the Balanoglossus. And in Asterias, it having the what that is a nerve ring around the mouth and stomach, and the what that is a this radial nerve cord. So these are comparable structures, and these in both the organisms, the nervous system is poorly developed, not well developed, like in the like the brain and the spinal cord, and the nerves and the ganglion, and it forms the epidermal nerve plexus. Then biochemically, if you observe the proteins and the phosphagens present in the Balanoglossus, they closely resemble those with the what echinoderms. Means those proteins and the phosphagens present in the Balanoglossus, these are chemically similar to those are found in the what echinoderms. And their common habit, that is both are the what exclusively marine, that is a echinoderms as well as the what hemichordate or Balanoglossus. These both are the exclusively marine. Then their ecological niches where they are found in the marine water and remarkable, and both are having the what great power of regeneration. These are the what the common similarities of the Balanoglossus with the Echinodermata. So we have discussed all the these five similarities of this what the Balanoglossus with the Echinodermata. Enterocolic origin of coelom. Then heart vesicle and glomerulus of Balanoglossus is homologous to the dorsal sac and axial gland of Echinoderm. Then the nervous system is poorly developed in both the organisms, that is Balanoglossus and Echinoderms. Then proteins and phosphagens present in Balanoglossus, they closely resemble or similar to the Echinoderm, and their common habit, that is they both are exclusively marine. Their ecological niches are very similar, and they having both that is the echinoderms as well as the hemichordates or Balanoglossus having great power of regeneration. So this is regarding the what adult similarities of the Balanoglossus with the what the Echinodermata. Then next, that is the what the larval resemblance or affinity of the Balanoglossus with Echinodermata. The what are the similarities with the larva? The larva of the Balanoglossus that we have already discussed. This is the Tornaria larva of the Balanoglossus, and we have also discussed the larva of the Echinodermata in last unit. That is the what this is the larva that is called as the bipinnaria. If you observe the what similarity between these two larvae, the Tornaria larva of Balanoglossus has striking structural similarities with the bipinnaria larva of the Echinoderms. These both the larvae are small, then pelagic, that are the surface feeder, transparent and oval in appearance. These are having the identical ciliated bands, that is a preoral and postoral ciliated band, and taking up the similar twisted course. And enterocolic, in both the coelom is how it is enterocolic type of the origin, and just we have discussed the what how the enterocolic origin coelom is there, and similar in the enterocolic origin and similar development of the coelom. So these are the what the larger larval resemblance, means the larva they resembles with each other, that is the Tornaria larva shows the similarity with the what that is a bipinnaria larva up to certain extent. Okay, next there is a dissimilarities in between the larvas also. Just we have discussed the what that is this larva, Tornaria larva. If you observe the of the what this Balanoglossus, the proboscis coelom, this Tornaria larva having the what that is a this proboscis coelom of the Tornaria, it is comparable to the hydrocele of the Echinoderm. This is the cavity. This is the what the hydrocele. This is the what the resemblance or the that is the similarity, not the dissimilarity. That is the similarity that is our Tornaria larva, the protosil. If you observe the what that is a coelom in the what that is the embryonic stages, this is the proboscis coelom, and this is the what the hydrocele of the what that is a diplural larva of the what echinoderm, and these are the comparable. So this is one of the again affinity or that is a similarity. And next similarity, that is a deuterostomes. Deuterostomes means what happens if they see you are having the what this is the development from the cleavage or the that is a what after fertilization, that is a what this. And the what happens here, that when the what the invagination occurs, there is a formation of the blastome. This blastopore is a opening, and inside it there is a cavity, that is a archenteron cavity, from where the gut will develop. Okay, so this blastopore, this is the opening. This opening either it develops into the mouth or anus. Okay, so those organisms where the what the blastopore or this opening it becomes a anus, these are called as a deuterostomes. And where this opening it develops into the what that is a mouth, these are called as a protostome. If you observe the what the development of this Tornaria larva and the what the development of this Echinoderm larva, in both these the deuterostome or these both are the deuterostomes. That is the blastopore becomes anus in Tornaria as well as these Echinoderms. And if you observe the digestive tract of this, the digestive tract is complete. That is what the mouth, then esophagus, then stomach, intestine, and what anus. Here also you can observe the what this mouth, then esophagus, then what the stomach and intestine, and the what that is anus. So all these both the larvas, that is the diplural larva as well as the what Tornaria larva, these having the complete digestive tract with mouth and anus along with the what esophagus, stomach, intestine, and the what this hydrocele, it is comparable to the what that is a proboscis coelom of the word Tornaria larva. So these are the what these six affinities or the adult affinities. So sorry, that is the larval resemblance or the larval affinities of the Balanoglossus with the Echinoderm larva. Then next, the dissimilarity in this. If you observe, this is a larva, Bipinnaria larva, and this is a Tornaria larva. Here you can observe the what the apical tuft of cilia. There is absence of apical tuft of cilia in the word this larva or the Bipinnaria larva. Then below this there is a eye spot. Here there is absence of the eye spot in the Bipinnaria larva. Then apical plate and the telotrope. This apical plate, this is the apical tuft. Then next, that is the what it having the what around the anus, it having the ciliary band, and that we called it as a telotrope. That is found in the what the Tornaria larva of the these Balanoglossus. But this telotrope is absent in the what the Bipinnaria. And the protocele, if you observe the word that is a protocele, this is a protocele means proboscis coelom. If you observe the proboscis coelom in the what this Tornaria or proboscis coelom during the development of the Balanoglossus, this is a what unpaired. This is a single. It remains as a what the single cavity, that is unpaired. While in echinoderms, if you observe the what this protocele, it is a what the paired. This is the what the developing coelom or that coelom is developed. That's a what that is a paired. So paired, that is a what protocele in the echinoderm, and unpaired protocele in the what the that is the Balanoglossus or the what the hemichordate. So these are the dissimilarities. So on the basis of the all the observations, that we come to the what that is a certain conclusions. The Tornaria larva shows the what the presence of apical plate with sensory hairs, then eye spots, telotrope, which are absent in the echinoderm larva. Then protocele is a single in Tornaria larva, just we have discussed, but paired in Echinoderm larva. This raises the doubt about the Echinoderm affinities of the Balanoglossus. Why? Because they are having the different, that is a what the coelom, that is it having the single coelom, or it is a single unpaired and the Echinoderm having the paired coelom. Then Fail in 1963 and others believe that their larval similarities are only because of convergent evolution due to same mode of habits and habitat. As they are what that is a sharing habit, same habit and habitat, there may be the what the larval similarities that was proposed by the what Fail in 1963 and other scientists. Inclusion of Balanoglossus under phylum Chordata is not universally accepted. Why? Because we have discussed the word that is the three fundamental characters of chordate. That is the nerve cord, notochord, and the gill slit. Out of these three important characteristics, only the word these Balanoglossus resembles remarkably with the gill slits or presence of gill slits. Okay. Why? Because the nerve cord, that is what it is present there, but it resembles to the what that is or the nervous system, it resembles with the other non-cortex. And the next structure, that is a notochord. The notochord is not true notochord in the Balanoglossus, but it is a what the buccal diverticulum. So among the three fundamental chordate characteristics, the nature of notochord is really questionable. The recent workers do not accept the notochordal nature of the buccal diverticulum because embryologically its origin is different from the notochord. Then the nervous system in general is a typically non-chordate type, excepting the presence of lumen in the collar nerve cord. The only important chordate feature is the word that is a gill slit, which strikingly similar with the word that is a chordate. So, however, recent DNA analysis, such as the word that is a 18R DNA analysis of entrepreneurs, or that is entrepreneurs as order, that entrepreneurs of the semichordata or Balanoglossus, this DNA analysis of entrepreneurs suggests that they are very closer to echinoderms than to the chordates, even though the morphologically they are very much different from the echinoderms, or in the what that is the body symmetry, they are very much different from the echinoderms, but the DNA analysis, it reveals that they are very close to the what that is the echinoderms than to the what the chordates or other groups. So by studying the what the affinities of these Balanoglossus with the what the echinodermata, chordate, and the annelida, we come to the conclusion that the above affinities, that echinoderms, hemichordates, and chordates have arising from the common ancestral larva. Means millions year ago there may be the what same larva or the same ancestral larva of all these three groups. That is the may be there of these echinoderm, hemichordates, and chordates. And echinoderm deviated greatly from ancestral stock and form a blind branch in the main line of evolution and continue to give rise to the hemichordates and chordates. So this was the what the how the what the characteristic features of the what that is the this Balanoglossus, which resembles with the Echinodermata, that is the what the larval characteristics as well as the what the adult characteristics. So these are the what the affinities or the resemblance, similarities and dissimilarities of the Balanoglossus with the phylum Echinodermata, adult resemblance, adult dissimilarities, then larval resemblance and the larval dissimilarities. So all these are the what regarding the what the affinities of Balanoglossus with the what the Echinodermata. Thank you.